morning, all of you. Today we will discuss about that is very easy topic, that is enzyme. In that we will see the introduction of enzymes, properties of enzymes, nomenclature, and IUP classification of enzymes. Let's start with introduction of enzymes. Enzymes, the biological catalyst that speed up the rate of biochemical reaction, is called enzymes. In that, most of the enzymes are three-dimensional globular proteins. They have tertiary and quaternary structure. Some special RNA species also act as enzymes and are called ribosomes. Example is hammerhead ribosomes. In our daily body, the various biochemical reactions occurs. In that, enzymes are very essential for the increasing the rate of activation of energy. Enzymes are also required. Next is properties of enzyme, that is characteristics of enzymes. In that, first is enzymes speed up the reaction by lowering the activation energy of reaction. Second, their presence does not affect the nature and properties of in product. Third, they are highly specific in their action, that is each enzyme can catalyze one kind of substrate. Third, small amount of enzymes can accelerate chemical reactions. Enzymes are sensitive to change in pH, temperature, and substrate concentration. Turnover number is defined as the number of substrate molecules transported per minute by one enzyme molecule. The turnover number is defined as the number of substrate molecule transported per minute by one enzyme molecule. The next, nomenclature. The nomenclature of enzyme, in that an enzyme is named according to the substrate name of the substrate it catalyzes. Some enzymes were named before a systematic way of naming enzyme was formed. Example, pepsin, trypsin, and renin. By by adding suffix ase at the end of the name of substrate, enzymes are named. Enzymes for catalyzing the hydrolysis is termed as hydrate. In that example, in that given below given is reaction. In that reaction, maltose plus water in presence of maltase enzyme, it form glucose plus glucose. In that reaction, maltase is act as enzyme. Next is in tribal name of enzyme, AAC suffix is used. According to IUBMB has developed an unambiguous system of enzyme nomenclature. IUBMB proposed a specific enzyme commission number to each and every enzyme. Each enzyme has a unique name and code number that identify the type of reaction catalyzed and the substrate involved. As per this system, the name start with EC, that is enzyme commission, followed by 4 digits. That 4 digits as follows. In that, first digit represents the class. In that, the first digit represents the class of enzyme. Second digit stands for subclass of enzyme. Second digit stands for subclass of enzyme. Next is third digit is subclass or subgroup. In next, fourth digit give the number of particular enzyme in the list. Fourth digit give the number of particular enzyme in the list. Next is classification. We will see classification of enzyme. Classification. A systematic classification of enzyme has been developed by International Enzyme Commission. International Enzyme Commission developed a systematic classification of enzyme. This classification is based on type of reaction catalyzed by the enzymes. The classification is based on type of reaction catalyzed by enzymes. There are six major classes. Each class is further divided into subclasses, subclasses, and so on to describe the huge number of different enzyme catalyzed reactions to IUBMB has developed. Each class, in that there are six major classes. Each class is further divided into subclasses, subclasses, and so on to describe the huge number of different enzyme catalyzed reactions to IUB has developed. Next, enzyme enzymes are classified into six functional classes. EC number classification, that is Enzyme Commission number classification by the International Union of Biochemists, that is short form is IUB, on the basis of types of reaction that they catalyze. In that there are seven classes. Last seven is newly added class in 2008. In that first is EC, that is Enzyme Commission number 1, that is oxidoreductase. Second, EC number 2, transferase. Third is EC number 3, hydrolysis. Fourth, EC number 4, lysase. Fifth, EC number, that is enzyme commission number 5, isomerase. Sixth is EC number 6, ligalase. Seventh, that is newly added, EC number 7, translocase. Next, hexokinase catalyzes the reaction belonging to the enzyme commission number 2, that is transferase. That is the given below the reaction. Glucose react with ATP in presence of hexokinase to produce glucose 6 phosphate plus ADP molecule. The reaction says glucose react with ATP in presence of hexokinase enzyme to produce glucose 6 phosphate plus ADP molecules. EC classification number 2.7.11, 2 belongs to the transferase class 7, and their next 7 alcohol involved in the reaction, that is OH group. One belongs phosphate group is transferred, and one stands for individual enzyme number. Systematic name of hexokinase is D-glucose 6 phosphotransferase. Here the lecture is over. Thank you for listening.